Hello everyone, my name is Dr. Yashoda Chaudhary and today I am going to take a previous year question session in which we will revise few of the PYTs in different subjects, and also you can find these PYQs and PYTs in the Limbus Med app and Limbus Med website. So you should definitely visit limbusmed.com, in which you will have access to PYT based notes, PYT based Q banks, mini test, everything which is based on PYTs, and PYTs are the ones which we have to master for any exam, whether for FMG, NEET PG or INSET. Okay, so without any further delay, let's discuss few of the question with little bit explanation, and let's revise. Okay, let's start with first question. Which of the following substances best used to measure extracellular fluid volume? So they are asking how to measure ECF. So first of all, we should know what is ECF. So this is a very basic physiology question. Whenever we start reading physiology, the first topic they will teach us is this topic. What is body fluid? है ना, basic. So let's just revise this basic body fluid physiology once. We all know that total body water is 60% of body weight. Yes, 60% of body weight is total body water, and total body water comprises of ECF and ICF. Okay, so that 60%, how it is made? Basically ICF is 40% and ECF is 20%. Okay, so see, this ECF is basically made up of plasma and interstitial fluid. Okay, so this is 20%. How this is 20%? See, plasma is 5% and interstitial fluid is 15%. Okay, so that's how our ECF is made. And see, ICF में मेजर आयंस कौन से होते हैं? ICF में मेजर आयंस जो होते हैं वो होता है पोटेशियम, दैन मैग्नीशियम, phosphate. Mostly these ions are present. Okay, and ECF में we have sodium, chloride and bicarbonate. So these are basic physiology things we should know before doing this question. Okay, so basically in this question they are asking ki how to measure ECF volume. Okay, to measure ECF the answer will be inulin. Okay, inulin is used to measure ECF. And what about other options? This D2O or sometimes T2O and also antipyrin. All these are used to measure total body water. Okay, and this Evans blue, it is used to measure plasma. Okay, antipyrin I told you that is total body water. That's it. So let's see. Yes, I told you that answer will be inulin, because inulin distributes only in the extracellular compartment and it does not enter the cells. Okay, that's why it is ideal for ECF measurement. D2 and antipyrin, they enter all the fluids compartment. D2O, T2O, antipyrin, all these enter all the fluid compartment, thus used to measure total body water. That I told you already. Okay, and Evans blue, basically this Evans blue, it binds to albumin in the plasma, and that's why it is ideal to measure plasma volume. Simple. Okay, let's see the next question. Which coenzyme is predominantly used in anabolic reaction? So first of all, what is anabolic reaction? Anabolic reaction are the ones जहाँ पे कुछ synthesis होता है, like something is made up, then that reaction is anabolic. And if something is breaking down, that is catabolic reaction. Okay, can you give me one example of catabolic reaction? And this is very basic question I'm asking. Catabolic reaction, that is beta oxidation of fatty acid. Yes, that is catabolic. Okay, so this is different. Now they're asking which coenzyme is predominantly used in anabolic reaction. मतलब synthesis में which coenzyme will be used. So I think, what do you think the answer will be? Like, is it NADH, FADH2, NADPH or coenzyme A? I think whenever this type of question come na, just think of ruling out other options. Okay, so NADH, where we are using this one, we are using in glycolysis, we are using in TCA, right? FADH2 also we are using in TCA, right? And coenzyme A basically we are using in fatty acid metabolism. So all these process, are they anabolic? No. TCA is both anabolic and catabolic basically. So the NADPH will be the answer, because NADPH we derive from HMP shunt, and further this NADPH used for synthesis of so many things like cholesterol synthesis, like steroid hormone synthesis, fatty acid synthesis. So yes, synthesis, everything is being synthesized, so that's why it is used in anabolic reaction. Okay, so okay, let's proceed with next question. Which stain best demonstrates basement membrane? Okay, so here the answer is pass. Okay, we'll see the options also. Why is the answer pass? Pass means periodic acid Schiff stain. Okay, see, basically basement membrane is made up of proteoglycans, glycoproteins. Okay, it is made up of type 4 collagen. It is made up of laminin, heparin sulfate. So basement membrane is made up of these structure, and all these are like basically carbohydrate based structure. Okay, so this pass na, it basically highlight the carbohydrate specific structures. So that's why pass is useful to stain the basement membrane. And the rest of options, just say HNE. HNE is not a specific stain. It's just used for routine screening. It's not specific stain. Okay. And GIMSA is mainly used for blood smears, bone marrow. Okay. Toluidine blue basically is a basic dye which is used to stain mast cells, cartilage. Okay, so yes, PAS highlights polysaccharides, mucosubstance and basement membrane. HNE is a general stain and GIMSA stain blood cells. Toluidine blue is for mast cell granules. Okay, next question is, which of the following marker is used to estimate intracellular fluid volume indirectly? So first of all, just remember one concept that we have studied earlier. Okay, we have ECF, we have ICF, and ECF comprises of plasma and interstitial fluid. So we have studied that we can measure total body water by D2O, T2O, antipyrin, that we have studied. We can measure plasma, we can measure ECF by inulin. Okay, that we have studied, but we have not studied about how to measure ICF, right? How to measure interstitial fluid, that we have not studied, because these cannot be measured directly. We have to measure these one indirectly, like we have to do efforts to measure these one. We just can't use one dye and measure this. Okay, so I and I, these initial we have to measure indirectly. And indirectly how? See, ICF we can measure by total body water minus ECF, and this interstitial fluid we can measure by ECF minus plasma. Okay, so we have to measure these indirectly. Okay, so let's see the option. Inulin and mannitol. Remember, this is for ECF. Okay, Evans blue is for plasma, and this tritium oxide, I told you T2O. Okay, this is for total body water. So first of all, we can just first measure total body water, ECF, and we can just do the minus thing. But also we can use tritium oxide indirectly, because basically T2O will enter into all the compartments, right? So we can use to measure ICF also. But inulin, mannitol and plasma we can't use, because inulin and mannitol will stay in the ECF compartment only, and that Evans blue will only stay in the plasma compartment. Okay, it will bind to albumin. So that's why the answer will be tritium oxide, because it enters all the compartment. Okay, so yes, tritium oxide distributes in total body water, and intracellular fluid is calculated as ICF equal to, I told you, total body water minus ECF. Okay, so it helps estimate ECF indirectly. Inulin measures ECF, I told you. Mannitol also ECF, I told you. And Evans blue only plasma. Okay, that's it. And let's see the next question. So which pathway is both anabolic and catabolic? Okay, see, we'll see option wise. First option is glycolysis. Is glycolysis is both anabolic and catabolic? See, both anabolic and catabolic means amphibolic. These pathways are known as amphibolic. Okay, so is glycolysis amphibolic pathway? I think glycolysis is breakdown of glucose now. It's just a catabolic pathway. Okay. What about gluconeogenesis? What you know about gluconeogenesis? Gluconeogenesis means synthesis of glucose from non-carb sources. Okay. Basically, it is synthesis of glucose. So, it is anabolic. So, both to nahi hai. It is not both. Okay. So, this is not the answer. This is not the answer. Urea cycle is just not amphibolic pathway. It is just a detoxification pathway for ammonia. Okay, so definitely this is not the answer. So answer is TCA cycle. Okay, TCA cycle is an amphibolic pathway. Amphibolic pathways are pathway that are both anabolic and catabolic. Okay, how TCA is amphibolic? Because TCA have both the role. TCA have anabolic role also, catabolic role also. Let's first see this. See, TCA cycle is amphibolic. It function in both breakdown of metabolites, and it provides intermediates for biosynthesis. What do you understand from this? See, TCA have anabolic role. What is the anabolic role? TCA intermediates. TCA intermediates, which are the TCA intermediates? Like citrate. Citrate is used for fatty acid synthesis. Okay, so this is anabolic role. Like succinyl CoA, used for heme synthesis. Okay, alpha ketoglutarate, used for amino acid synthesis. So basically this is anabolic role. And what is catabolic role for the TCA? Catabolic role means TCA basically break down or oxidize acetyl CoA. Okay, acetyl CoA oxidization, and also it produce NADH, FADH2, GTP. Okay, so that is catabolic role, and anabolic role is that we have seen. Okay. Next question, selenocysteine is incorporated into protein via which codon? So they're asking the codon for selenocysteine. So yes, we have to remember this factual question. So the answer is UGA. Okay, UGA. Okay, so selenocysteine. Basically, UGA is also a stop codon, but it is incorporated into proteins for selenocysteine. Okay, so UAG is basically for pyrrolysine. Okay, pyrrolysine. UAA is just a stop codon. AUG we all know AUG. It is for methionine. You all know that AUG, it initiates translation. Okay, so methionine and initiates translation. I think have to, yes, it's initiate, translation, and AUG is a start codon for methionine. Okay, so yes, let's see. The answer is UGA recoding to selenocysteine. UGA recoding to selenocysteine is a unique code translation process. So yes, A and D are unrelated and B occur after translation, not during. Okay, so basically just have to remember that UAG is for pyrrolysine, UGA is for selenocysteine, UAA is just a stop codon, AUG is for methionine. It is a start codon. Okay, it initiates translation. That we have to remember for this question. Okay, next question is, which of the following methods would best allow a clinician to determine plasma volume? I think by now you should just having this one on your tips. We have revised it like 1 or 2 times. Okay, so Evans blue will be the answer. Plasma Evans blue, simple. Okay, so let's see. Inulin, we all know inulin ECF. Tritium is for total body water and it is for plasma. Okay, so yeah, Evans blue binds to plasma, it binds to albumin, and it stays within the vascular compartments, and it making it ideal for plasma volume measurement. Inulin, we all know, is CF. Okay, and radioactive sodium also measure ECF, and it distributes into interstitial fluid. Okay, so that's it. Which of the following sugar will test positive with Benedict's test? So what is Benedict's test? It is a test for reducing sugars. Okay, to detect reducing sugars. So I think it's very simple. We can just rule out, is sucrose a reducing sugar? Sucrose is a non-reducing disaccharide. Okay, fructose is a reducing monosaccharide. Okay, and cellulose and starch are basically polysaccharides. So the answer is, and see, cellulose and starch are very large molecule, so reducing is not possible. Like there is no reducing ends. Okay, so the answer is fructose. Benedict's test is for fructose, and which is a reducing sugar. This is the concept we have to remember for this question. See, fructose is a reducing sugar due to its ability to tautomerize to glucose and mannose. Sucrose is a non-reducing, and C and D are polysaccharide. And basically these reducing sugar, na, what are these? They have free aldehyde ketone group, na, they have free aldehyde and ketone group, and they further reduce copper 2 plus into copper. Okay, copper 2 plus into copper plus. Okay, so this is the concept of reducing sugar. We just have to remember this one. And do you know what test we use to detect starch? This is very basic. Iodine test, na. Okay, let's see the next question. The substance used to estimate total body water must have which of the following properties? So let's see. Should bind to plasma protein? No. Rule out. Plasma protein? No. Tritium oxide distribution? Maybe, because we have studied T2O, D2O, antipyrine for the total body water measurement. Evans blue is for plasma, just cut it. Radioactive sodium we have read for ECF. So yes, the answer is tritium oxide distribution, that we have discussed it, and you just have this topic on your tips now. Okay, so yes, total body water include ICF and ECF, so the substance must freely cross the cell membrane and distributes into all the compartments. But plasma protein binding restricts to plasma. That's why Evans blue, it binds to albumin only. Okay, so and yes, so next question is, which of the following stain is most specific for identifying amyloid deposits in tissue? So first of all, what is amyloid? Amyloid is basically extracellular beta plated sheet protein. Okay, it is an extracellular protein, beta plated sheet structure. So how to detect it is basically why, and we can just rule out. Okay, past we have studied for basement membrane, right? First just try to rule out how we can just rule out and just find our answer. Trichrome, I think it is related to some color or something. Trichrome. Okay, it will not be for amyloid, it will not be for amyloid definitely. Okay, Sudan black B is for lipid. See, trichrome is basically for collagen, it differentiate collagen and muscles. Okay, that is trichrome. So collagen and muscle को differentiate करने के लिए we use this trichrome stain. So the answer will be Congo red. See, Congo red basically it detects this beta plated sheet structure. That's why it is specifically used for amyloid deposits. Okay, so basically it give this apple green birefringes in the polarized light. So just you have to remember this keyword for this question. Okay, so Congo red is the gold standard stain for detecting amyloid, showing apple green birefringes under the polarized light. And PAS, we know it's stain glycogen, mucopolysaccharides, and basically for basement membrane, but not specifically amyloid. And trichrome stains connective tissues, and Sudan black B stains lipids. Lipids basically we can use for bone marrow, leukemias, and all these. Okay, so see, next question is which sirtuins is primarily localized to the mitochondria and regulate mitochondrial metabolism. So they're asking about sirtuins which regulate mitochondrial metabolism. Okay, so basically what are sirtuins? Just remember one short thing. They are, key regulator for anti-aging, metabolism, stress resistance. Okay, they are key regulator of metabolism, stress resistance, anti-aging, and basically they increase our lifespan. So in this question they are just asking ki mitochondria mein which sirtuins is available. So this is very factual question and we just have to remember this one. And basically this is a INI-set pattern question. Okay, so the answer is SIRT3 is in mitochondria, because SIRT1 is nuclear, 6 is nuclear, 2 is basically cytosolic. Okay, so 6 nuclear, 1 is in nuclear and cytoplasmic, 2 is cytosolic, and this 3 is mitochondrial sirtuin. It regulate oxidative metabolism and reactive oxygen species detoxification. Okay, so that's what you remember about sirtuins. And also remember how to increase sirtuins. Basically we can increase sirtuins by calorie restrictions, and also red wine consumption is linked to increase sirtuins. So basically increasing sirtuins means increasing our lifespan. Okay, that's it. Next question, which growth factor is primarily responsible for stimulating fibroblast proliferation and collagen synthesis in wound healing? So, fibroblast proliferation and collagen synthesis. Answer will be, just try to rule out. Now, have you ever read that VEGF is linked to fibroblast proliferation? No. PDGF? No. Platelet derived growth factor? No. EGF, epithelial growth factor? No. So, answer is TGF beta. See, TGF beta helps in wound healing by fibroblast proliferation. It increases collagen synthesis. It increases collagen synthesis, and basically which collagen? Type 1. Okay, type 1 more than 2. And finally it helps in scar formation and wound healing. Okay, so this is a concept for TGF beta. So TGF beta promotes fibroblast proliferation, collagen deposition, wound remodeling. And what about VEGF? VEGF promotes angiogenesis, new blood vessels. Okay, PDGF it recruits inflammatory cells, and EGF stimulates epithelial regeneration. Next question is, prozone phenomena is most commonly associated with which type of serological test? So this is a micro question. Answer is agglutination test. Okay, let's just see this one in brief. What is prozone phenomena? It's basically a false negative reaction. Okay, which reaction? Agglutination reaction. Means they're just asking ki prozone phenomena is associated with which test. So agglutination. So basically what is agglutination? Agglutination means we check antigen and antibody interaction, right? So what happened in prozone phenomena? There are excess antibodies. Excess antibody, that means there is no proper antigen and antibody interaction, and there is no proper lattice formation, okay, which they called as lattice theory. When there is optimum antigen and antibody interaction, there is lattice formation. So that is basically proper agglutination reaction. Okay, but prozone phenomena, यहाँ क्या हो रहा है? There is excess antibody and that's why there is no proper lattice formation. That's why agglutination test appears to be false negative. That is prozone phenomena. And there is also one post-zone phenomena. So post-zone phenomena means when there are excess antigen. So condition is same, excess antigen, that means there will be no proper antigen and antibody interaction. And if there is excess antibody, that is prozone. So there is also no proper antigen antibody interaction. So that is associated with agglutination test. Okay, so the answer is agglutination test. Prozone phenomena is typically seen in agglutination test. It is not seen in ELISA and precipitation of Western blot. No such phenomenon is seen. Okay, so which of the following is not an anaerobic infection? They're asking which is not an anaerobic infection. So let's just try to rule out. Tetanus. What do you know about tetanus? Tetanus produce tetanospasmin toxin. Okay, we all know about this. We all know that it is an obligate anaerobe. Okay, tetanus is an obligate anaerobe. Botulism, sorry. See, botulism is caused by Clostridium botulinum. We all know that. So it is also anaerobe. So obligate anaerobe. Necrotizing fasciitis is caused by Clostridium perfringens, which also cause like gas gangrene reaction, and it is basically a mixed infection of aerobic and anaerobic flora. So it is a mixed infection. Okay, so typhoid fever is caused by Salmonella typhi, and which is a facultative anaerobe. So let's just see the explanation. Typhoid fever is caused by Salmonella typhi. It is a facultative anaerobe, not an obligate anaerobe. Okay, so you must be confused ki what is difference between facultative anaerobe and obligative anaerobe. See, obligative anaerobes are the one which will grow only in the absence of oxygen. Okay, their growth strictly depends on the absence of oxygen, but facultative anaerobes, they can grow with or without oxygen. Okay, so that's it for this question. So next question is, pH indicator used in clad medium is? So clad medium basically we use mostly for urine cultures. Okay, so they are asking for the pH indicator. So answer is bromothymol blue. Okay, so basically it differentiate lactose fermenter versus non-lactose fermenters. So if they are lactose fermenters, it will produce yellow colonies. Non-lactose fermenters, it will produce blue-green colonies. Okay, and rest of all, जैसे like phenol red, it is used for mannitol salt agar. Okay, and neutral red is for McConkey agar. So that's it you have to remember for this. See, bromothymol blue. Bromothymol blue is a pH indicator in the clad medium, turning yellow in lactose fermenters. Okay, and rest of others we have seen. Okay, which antiemetic is a muscarinic M1 receptor antagonist effective for motion sickness? Okay, so options are scopolamine, ondansetron, aprepitant, and prochlorperazine. Okay, so the answer will be, let's just rule out. Okay, ondansetron, we all know it is 5-HT3 blocker. Aprepitant is a repeated asked question. It is NK1 blocker, okay, antagonist. NK1 antagonist. Prochlorperazine is a D2 blocker, okay. So answer will be scopolamine, okay. Scopolamine blocks M1 receptors in the vestibular nuclei. It reduces motion induced MSS, okay. So ondansetron is mostly for chemotherapy induced vomiting, we all know. Okay, aprepitant is also for chemotherapy. If they ask early chemotherapy induced vomiting, then it is ondansetron. Late, then it is mostly aprepitant, okay. And that is D2 blocker, prochlorperazine. Which color do the metachromatic granules appear in the Albert stain? So Albert stain basically we use for diphtheria, okay, Corynebacterium diphtheria. That is a repeated question. So they're asking metachromatic granules color. So the answer is black color, okay, not blue color. Answer is black color. So basically metachromatic granules appear black against a green background, okay. So that is for diphtheria. See, next question is which antiemetic can cause extrapyramidal side effect due to central dopamine receptor blockade, especially in young patients. So they're asking about dopamine receptor blockade. So we can just rule out dexamethasone is steroid. Just rule out. Aprepitant we have studied, it was NK1 blocker, okay, and NK1 antagonist. So it is ruled out. So ondansetron we have studied, so it is also ruled out. Answer will be metachlorbromide. First just rule out and find out the answer, and let's just discuss metachlorbromide, okay. See, this drug, it is basically antiemetic drug and also prokinetic drug. Why it is prokinetic? Antiemetic because it block dopamine in the CTZ area, okay, that's it. But why it is prokinetic? Because it is also 5-HT4 agonist. So basically 5-HT4, what it do, it increase motility in our gut. So this drug, it is a agonist for 5-HT4, so basically it is a prokinetic drug and antiemetic, that's it. But why it is causing uh, this extrapyramidal side effect? Because it also block dopamine in the basal ganglia. Basically the dopamine in the basal ganglia responsible for smoothing of voluntary movement, and if you are blocking the dopamine, that leads to acute dystonia, akathisia, tardive dyskinesia and all that. So that's it. You have to remember that metachlorbromide is antiemetic plus prokinetic drugs. Antiemetic, it blocks dopamine in the CTZ area and it also blocks dopamine in the basal ganglia, and that's why it can lead to extrapyramidal side effects. And prokinetic drug because it is 5-HT4 agonist, okay. So, which PPI interaction can reduce the efficacy of clopidogrel by competitive metabolism? See, first of all, I want to say that this question, this topic, they have repeated so many times in all the exams. Whether in NEET, PG, FMG, INS, at every exam, they want to ask this topic by either like they'll ask anyway, but they will ask. Don't know why they love this question so much, but let's see the concept. See, clopidogrel mostly it is given to CAD patients, okay. It is an antiplatelet drug, that we know, it's basic. See, which PPI interaction can reduce its efficacy? So basically, if someone is taking clopidogrel and also taking one PPI drug along with it, maybe he must have some acidity issue, so he must be taking some PPI along with it. So that is omeprazole. If he is taking omeprazole and clopidogrel together, that means clopidogrel will not work, because clopidogrel is activated by a CYP enzyme which is CYP2C19, okay. So let's see the concepts. CYP2C19, it activates clopidogrel, okay, but omeprazole, it inhibits CYP2C19 only. So if you are giving omeprazole and clopidogrel together, omeprazole will inhibit the CYP2C19, and further CYP2C19 will not able to activate the clopidogrel. Okay, so that's why clopidogrel will not work. So CYP2C19, it reduces clopidogrel activation. That's why they should not be given together. So pantoprazole and rabeprazole have minimum CYP2C19 inhibition, okay, and rabeprazole less CYP interaction, okay, and esomeprazole also inhibit but to lesser degree than omeprazole. But mainly you have to remember omeprazole, because omeprazole inhibits CYP2C19, which was activating clopidogrel. Okay, just remember this concept. They have asked a lot of times. Okay, next question is, which anticoagulant directly activates antithrombin 3, okay, to inhibit both factor 10A and 2A. Okay, so which anticoagulant? The answer is unfractionated heparin, UFH. Okay, let's see the option. See, UFH, it binds to antithrombin and it accelerates inhibition of thrombin and factor 10A. Thrombin is basically factor 2. Okay, so this unfractionated heparin, it blocks both factor 2 and factor 10. Okay, but the fondaparinux, it blocks only factor 10. Just remember this one. Okay, they sometimes ask the difference between these two. So UFH both 2 and 10, but fondaparinux only block factor 10. Okay, and bevalirudin is a direct thrombin inhibitor. Okay, this bevalirudin and argatropan both are direct thrombin inhibitors. Okay, so that's it. These topics, these questions have been asked multiple times. We have to remember these topics, these questions, because they can be repeated in another way in any exam. So keep revising these questions, and you can just have access to these topics and their explanations in LimbusMed app and LimbusMed website. So just keep learning. Thank you.